Hello. 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 Okay. Can you hear me, Chirant? Chirant? Tejaswini? Can you hear me? Can you hear me? Hello. Can you hear me?
Yes, sir.
Very good. What about Tejaswini? Tejaswini, am I audible to you? So Chirant spoke. What about Tejaswini? I don't know. What about Vijay? Vijay is joining. Tejaswini, I cannot hear your voice. Chirant, can you interact with me? Chirant,
Yes, sir.
is the voice clear? Is the voice clear to you?
ಸ್ವಲ್ಪ ಕಟ್ ಕಟ್ ಆಗ್ತಾ ಇದೆ ಸರ್ ವಾಯ್ಸ್.
ಕಟ್ ಆಗ್ತದೆ. ಇನ್ಯಾವ ಆ್ಯಪ್ ಯೂಸ್ ಮಾಡೋಣಪ್ಪ? ಐ ಥಿಂಕ್ ಇಟ್ಸ್ ನಾಟ್ ಎ ಪ್ರಾಬ್ಲಮ್. ನೋಡು, ನೆಟ್ ಏನೋ ಪ್ರಾಬ್ಲಮ್ ಇರುತ್ತೆ. ಇಟ್ಸ್ ಅನ್ ಇಶ್ಯೂ ವಿತ್ ದಿ ನೆಟ್. ನೆಟ್ ಪ್ರಾಬ್ಲಮ್ ಆದ್ರೆ ಎವ್ರಿಥಿಂಗ್ ವಿಲ್ ಬಿ ಎ ಪ್ರಾಬ್ಲಮ್. ಯು ಶುಡ್ ಹ್ಯಾವ್ ಎ ಗುಡ್ ಕನೆಕ್ಟಿವಿಟಿ.
Yes, sir.
Okay, so see that you are well connected to
Yes,
the
sir.
internet. Then only you can hear voice and the video, right? Okay. 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 So shall I start? Shall I start? 4 students have joined. So Manikanta has joined. Manikanta, can you hear me? ಕೇಳಿಸ್ತಾ ಇದೆಯಪ್ಪ ಮಣಿಕಂಠ? ಮಣಿಕಂಠ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ? ಮಣಿಕಂಠ,
ಸರ್ ಕೇಳಿಸ್ತಾನೆ ಸರ್?
ಹಾ, ವೆರಿ ಗುಡ್. ವಿಡಿಯೋ ಕಾಣಿಸ್ತಾ ಇಲ್ವಾ? Video.
Sir.
Okay. Hello. Hello, Prashant. Prashant is logging in. Okay. There are 5 students who joined. Okay. Tejaswini, can you hear me?
Yes, yes.
Can you hear me? Okay, fine.
Yes, sir.
Chirant, Manikanta, all of you.
Yes, sir.
Okay.
Yes, sir. Yes, sir.
Okay. Shall I start sharing the PPT now? So let me share the screen.
Sir, ನನ್ನ ಹೆಸರು ಕರೀಲಿಲ್ಲ.
ಯಾರಪ್ಪ?
ಮಣಿಕಂಠ.
ಮಣಿಕಂಠ ನಾ ಕರೆದ್ನಲ್ಲೋ ಎಷ್ಟು ಸರಿ ಕರೆದ್ನೋ 2-3 ಸರಿ ಕರೆದ್ನೋ.
ಹೌದಾ? ಸರಿ ಸರ್, ಸರಿ ಸರ್ ಆಯ್ತು. ಸರಿ ಸರ್ ಆಯ್ತು.
ಇನ್ನೊಂದು 5 ಸರಿ ಕರೆದು ಬಿಡ್ತೀನಿ ಬಿಡಾತಗೆ. ಮಣಿಕಂಠ ಮಣಿಕಂಠ ನಿನ್ನ ಅಯ್ಯಪ್ಪನ
ಬಿಡಿ ಸರ್, ಸಾರಿ.
ಸರಿ ಬಿಡು.
ಇದು ಫಸ್ಟ್ ಟೈಮ್ ಬಂದಿರೋದು, ಅದಕ್ಕೋಸ್ಕರ ಸರ್.
ಹೌದೇನಾ? ಹೋಗ್ಲಿ ಬಿಡ. ತೊಂದ್ರೆ ಇಲ್ಲ. ಇನ್ನೊಂದು 5 ಸರಿ ಜೈಕಾರ ಹಾಕ್ಬಿಡ್ತೀನಿ. ಹೆಂಗಿದ್ರು ಮಣಿಕಂಠ ಅಂತ ಇಟ್ಕೊಂಡಿದೀಯ. ಅಯ್ಯಪ್ಪನ ಹೆಸರು ಹೇಳದಂಗೆ ಆಯ್ತದೆ. Okay, let me share the PPT. You go through the PPT and we will have a discussion at the end of this class. Okay, shall I start sharing the PPT? So let me start. Still there are some more students joining. Okay, Somadarshan has joined. Prashant has joined. So there are 5 students as of now who have logged in the class. Okay. So let me share the PPT. Okay. So let me share. Or shall we wait for 2 more minutes? We'll wait up to 12:40. Okay. Because only 40 minutes is the presentation time. I hope so. If it runs for 1 hour, we could have waited. As the class runs for only 40 minutes, we will start at 12:40. Okay. Somadarshan, you are speaking? Okay. Okay, fine. So am I audible to you? Am I audible to all the participants? Am I audible to all the participants? ಕೇಳಿಸ್ತಾ ಇದೆಯಾ ಎಲ್ಲರಿಗೂ? ಪ್ರಶಾಂತ್
ಸರ್, ನಿಮ್ ವಾಯ್ಸ್ ಬ್ರೇಕ್ ಆಗ್ತಾ ಇದೆ, ಸರಿ ಕೇಳಿಸ್ತಾ ಇಲ್ಲ.
ಇದೆ ಏನೋ ನೆಟ್ವರ್ಕ್ ಇಶ್ಯೂ
ಸರ್, ವಾಯ್ಸ್ ಬ್ರೇಕ್ ಆಯ್ತೈತೆ ಸರ್.
ಹೌದೇನಾ? ಅದೇನೋ ನೆಟ್ವರ್ಕ್ ಇಲ್ಲಪ್ಪ. ಇನ್ನೊಂದು ನಿಮಿಷ, ನೆಟ್ ಏನಾದ್ರು ಪ್ರಾಬ್ಲಮ್ ಇದೆಯಾ ನೋಡ್ತೀನಿ ಒಂದ್ಸರಿ. If it is a network issue.
ವಿಡಿಯೋ ಬ್ಲರ್ ಆಯ್ತೈತೆ ಸರ್.
ಹೌದೇನಾ? ಈಗ is there any network issue? ನಮ್ದು ನೆಟ್ವರ್ಕ್ ಪ್ರಾಬ್ಲಮ್ ಆದ್ರೆ ಹಂಗಾಗತ್ತೆ. ಈಗ ಆಗ್ಬೋದಾ? ಬರ್ತಾ ಇದೆಯಾ? ಓಕೆ, ಕೇಳಿಸ್ತಾ ಇದೆಯಲ್ಲ?
ಈಗ ಸ್ಪೀಕರ್ ಆಫ್ ಮಾಡಿದೆ. ಈಗ
So let me start sharing the PPT now. I hope I can okay, Sneha. Okay. Okay, shall I start? Chiran, can you hear me? Chiran, ಕೇಳಿಸ್ತಾ ಇದೆಯೇನಪ್ಪ?
ಹಾ ಸರ್, ಕೇಳಿಸ್ತದೆ.
ಪ್ರೆಸೆಂಟ್ ಮಾಡ್ಲಾಗದ್ರೆ PPT ಇನ್ನು ಜಾಯಿನ್ ಆಗ್ತಾನೆ ಇದಾರೆ. ಈಗ 1 2 3 ಆಗಿದ್ದಾರೆ ಸಾಕು. 1 2 3 4 5 6 7 ಜನ ಆಗಿದ್ದಾರೆ. ಸ್ಟಾರ್ಟ್. ಇನ್ನು ಅರ್ಧ ಗಂಟೆ ಸಿಗುತ್ತೆ ನಮ್ಗೆ ಅಷ್ಟೇ. ಅದಕ್ಕೆ ಓಕೆನಾ? So shall I start? Participant to unmute themselves. So current and new participants will be muted. Mute. Okay, shall I start? So let me start. Okay. So let me start. Okay. Can you see PPT? Presented? Can you see PPT? All of you. Can you see the PPT?
ಸರ್ ನಿಮ್ ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇಲ್ಲ ಸರ್.
ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇಲ್ವೇನಪ್ಪ? ಏನಪ್ಪ ಮಾಡೋದೀಗ? What can I do? Can you see the PPT?
No sir.
You cannot see
No,
the
sir.
PPT. Okay, wait a minute. This is a new app for me. Even I don't know how to share. Share. I have given. Okay, fine. Share. Okay. Yes. Share. I think now I am able to. Now can you see? I think you can see now.
Yes, sir.
Okay. So I'll proceed with the PPT. Okay. Wherever you get doubts, you can interact with me. Okay. So let me go because it's already 11:45. We have to finish off in another 30 minutes. Okay. So coming to the history of viruses, can you see all of you? Can you see the slide?
Yes, sir.
Okay,
Yes sir.
so go through the slide. So it started all with the plant diseases, history of viruses I am talking. So the first disease was breaking of tulip. Tulip is a vegetable, right? It was recorded as early as in 1576. That was the time when the viral disease was reported in tulip. So later on, potato curl disease and peach yellows were also recorded respectively in the year 1765 and in the year 1791. So this was the time of the plant disease, record of the plant diseases, right? But the people were not knowing that it is due to the viruses, but these diseases were there before, right? Did you understand? Did you understand, participants? Did you follow? Did you
Yes
follow?
sir.
Okay, fine. So around 1868, variegation in abutilon was noted, right? Its variegation means some change in the color of the shoot or in the leaf or in any part of the plant was noticed. That was in 1868, and in 1870, a crop, tobacco crop, was affected, right? So it was Adolf Meyer was the director of agricultural experiment station, began his studies on this disease. So this became curious for Adolf Meyer. So he was the one who started working on tobacco crop. So there were diseases in other crops, but his focus was mainly on the tobacco crop, right? So it was Adolf Meyer who for the first time started working on the tobacco crop and its disease, okay, and published his results also. So he started working on it and he published his findings in the year 1886. So later on, it was Meyer named the disease as mosaic cranket. So the disease of tobacco crop was named as mosaic cranket, which means mosaic. So what is mosaic means? Can anybody define me what a mosaic is? What mosaic is? What is mosaic? What is mosaic? You have studied this before. Can anybody define me what mosaic is? What is mosaic? No answers. Chirant, Tejaswini, any answers from you? No answers. Chirant?
No, sir.
No. So mosaic means it is an alternating patches of green and white. So green and white patches were seen on the leaf. right? So there was a dark green patch followed by the light green patch. So that formed a mosaic on the leaf. So this disease was prominent on the leaf surface. You know that, right? So therefore, this was called as mosaic. Okay. So he was able to demonstrate the infectious nature of the sap of the diseased leaves. So the sap means the liquid of the diseased plant could be collected, and when it can be filtered also. It could pass through the double filters. When that sap was allowed to pass through the filters, right? So that's how the work was started. They found that there were mosaics and they could extract the sap from the infected crop. Okay, sap is the liquid, okay, from the diseased leaves. They could collect the sap of the diseased leaves. Okay, it is therefore considered that the science of viruses or virology was born in 1886. So you have to remember this year. So this is the time. This was the time of origin of viruses or virology. So this was born in the year. This was born in the year. Virology born in the year. When it was born? When it was actually started? When the virology started? When the virology started?
1886, sir.
1886.
Very good, very good. In the year 1886, the virology started with the work of which crop? With the work of which crop?
Tobacco.
Tobacco. Fine. However, later on, it was Russian scientist named Ivanovsky. In the year 1892, he demonstrated that the tobacco mosaic could be transmitted to healthy plants. So he was the one who found that the sap is transmissible. It can be transferred. So the diseased plant can make other healthy plants diseased. Okay. So whenever the sap is applied on the healthy plants were also infected. So that was his finding. So Ivanovsky was the one who demonstrated the transmissible nature of the sap. Is this clear? Okay. In addition to that, Beijerinck from Holland also found that the infectious juice or the sap, now we came to know that there is a sap in the diseased plant. That sap is transferred. It can transmit the disease to the healthy plant, and that sap was called as infectious juice by the Beijerinck. So it remained infective even after passing through the bacterial filter. This was another important findings in the history of virology. So what he found was, Beijerinck, what he found was, these juices can be filtered through the bacterial filters and remain sterile. Did you understand? So what is the meaning of that? It can pass through the bacterial filters. What does it mean? This juice can pass through the bacterial filters. What does it mean? ಏನು ಅರ್ಥ ಆಗುತ್ತೆ ಇದರಿಂದ? ಏನರ್ಥ ಆಗುತ್ತಪ್ಪ? ಸೋಮದರ್ಶನ ಏನರ್ಥ ಆಗುತ್ತೆ ಇದರಿಂದ? What do you understand from this? Anybody?
Sir, infective ಆಗಿ ರಿಮೈನ್ ಆಗುತ್ತೆ ಸರ್.
ಏನರ್ಥ ಆಗುತ್ತೆ ಈ ಸೆಂಟೆನ್ಸ್ ಇಂದ? ಏನರ್ಥ ಆಗುತ್ತೆ? What do you understand from this sentence?
ಸರ್, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಫಿಲ್ಟರ್ ಮಾಡಿದ್ರು ಸಹ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿರುತ್ತೆ ಸರ್.
ಇನ್ಫೆಕ್ಟಿವಿಟಿ
ಫಿಲ್ಟರ್ ಮಾಡಿದ್ರೆ ಅದು ಫಿಲ್ಟರ್ ಆಗಲ್ಲ ಅಂತ.
ದಟ್ ಈಸ್ ಇದರಿಂದ ಏನು ಗೊತ್ತಾಗ್ತಾ ಇದೆ ಅಂತಂದ್ರೆ, ಸಿ ದಿಸ್ ಸ್ಯಾಪ್ ಹ್ಯಾಸ್ some material which is smaller than the bacteria. ಅರ್ಥ ಆಗ್ತಾ ಇದೆಯಾ? ಈ ಸ್ಯಾಪ್ ಅಲ್ಲಿ ಬ್ಯಾಕ್ಟೀರಿಯಾಕ್ಕಿಂತ ಚಿಕ್ಕದಿರುವಂತಹ ಏನೋ ಇನ್ಫೆಕ್ಟಿವ್ ಮೆಟೀರಿಯಲ್ ಇದೆ ಅನ್ನೋದು ಅರ್ಥ ಆಗಲ್ವಾ? ಬಿಕಾಸ್ when something passes through the bacterial filter, it means they are smaller than the bacterium, right? That is the meaning. The viruses which was not known earlier, this can be passed through the bacterial filters antandre they are smaller than bacterial filters tane. That is another thing that can be noticed from this. Okay, So these bacteria can pass through the bacterial filters. So he recognized the existence of new type of infectious agent and he called it as Contagium vivum fluidum. So this is an important word which means infectious living fluid. Infectious living fluid. So this was the term used by Byjernik. Remember this? Byjernik was the one who filtered it through the bacterial filters and he gave the name to this infectious agent as Contagium vivum fluidum. Subsequently, he referred this infective agent of tobacco mosaic disease as viruses. So till the work of Byjernik, we were not knowing the disease causing agent. So only after the work of Byjernik, we came to know that the disease causing agent is a virus. Did you understand? Though many viral diseases were reported before, either the animal diseases or the plant diseases, it is only with the work of Byjernik we came to know that the infectious agent of the tobacco were viruses. Did you understand? Okay. So almost concurrently, so almost at the same time, Frederick Loeffler and Paul Frost in Germany, utilizing a similar approach, concluded that the agent causing foot and mouth disease of cattle also passed through the porcelain filters. At the same time, people were working on the animal diseases also. So far I have discussed about the plant diseases, right? So the people, virologists, they were working on animal diseases also, and they came out with the same findings that they can also be filtered. The disease causing agent in the foot and mouth disease of cattle can also be filtered. Okay. This was another important observation to be made. So these observations, highly controversial at the time, provided the basis for defining viruses as subcellular entities. So this gave us evidence that the viruses are subcellular, which means they are smaller than the filters. They are smaller than the bacterium. So that is how we came to know that the viruses are subcellular entities. It's smaller than the bacterium because they can be easily filtered through the filters, bacterial filters. So the animal diseases, the work on the animal diseases, the work on the plant diseases gave an important proof on the existence of the viruses. So uniqueness of this detective work is even more dramatic when one considers that the infectious virus particles were too small to see and could not grow on the cultural media available. So there was another finding made that these small infectious agents cannot be grown on a medium. That was also one of the important findings if you look into the history of virology. Now, visualization of viruses avoided the use of electron microscope. Now they could say that there are some infectious agents that are causing the disease, but they could not see it. It was invisible, right? Because why it was not visible? Why the viruses were not visible? Why, Chiran? Why the viruses were not visible till 1930?
Very small.
See, an ordinary microscope with a limited magnification, it cannot magnify the viruses. So we needed an instrument which could magnify an object to a greater extent, right? So in the 1930s, people developed electron microscope, and that's how they could visualize the viruses. So before that, as the electron microscope was not discovered, the existence of viruses was not known directly. Their picture was not known directly. It's only the indirect effect. It is the indirect effect of the viruses that were known. What was the indirect effect? Indirect effect was in the form of diseases. So viruses, they were causing diseases. That was the thing which was known earlier. But with the development of the electron microscope in 1930s, we could visualize them, we could see them, we can take pictures of it, and so now we know that they are rod shaped. See, they have a different structure, right? With the discovery of the electron microscope, we came to know that the viruses have a particular structure. So, and it developed over the years, and in the year 1940s and 1950s, they were able to see the viral applications also. So the filter passage infected material from scrapie infected sheep caused a similar disease when inoculated back into healthy sheep. And they, at the same time, they also found that some filter passages from the scrapie infected sheep. Scrapie is a disease. So that inoculum, that filtered uh, passage, when it was inoculated into healthy sheep, also got the disease. So in the year 1980 and 1990, so this transmissible material was found to be prions. Okay, so they went ahead. So now you know that this is the time of prions and viroids. They have worked on the viruses. They have come to even smaller than the viruses. They have come to the level of the prions, right? So in the 1990s, they found that another disease was there in scrapie sheep, right? Sorry, in the sheeps, and that was scrapie, and that disease was due to another infectious material which was even smaller than the viruses, and that was termed as PRPs or prion proteins. It's shortly called PRPCs. Okay, this was the history. So these slides will tell us about the history of the viruses. Now we will go to the discovery of viruses, how the discovery was done, how the discovery was made. So it was in the tobacco crop, and it was Adolf Meyer, you all know that, who was the director of the Agricultural Experimental Station, who was the one who was studying the disease, mosaic disease. And in the year 1880, he was working on this disease, and he published his results in the year 1886. And Meyer, it was Adolf Meyer who coined, Christian means coined the term mosaic Karen-Keat, that is mosaic-like. He referred them as the mosaic-like, from the mosaic-like pattern on leaves. So there were mosaic-like patterns on the leaves of the diseased plants. Healthy plants did not show that symptom, but the diseased plants had these mosaic patterns. Okay, and he succeeded in reproducing the disease by infecting the Jews. So he also was successful in transmitting the disease from the disease to the healthy ones. Okay, so he could succeed in identifying the real cause of the disease. That was the time. Okay, so Mayer's contribution will always be remembered, as he was the first person who put first step forward in the development of new discipline, later recognized as virology. So therefore, we still remember Adolf Meyer for his discovery of viruses. So therefore, the virology itself started with the work of Meyer. He was the one who put a base, who put a platform to the discipline of virology, right? So the development of porcelain bacterial filters by Chamberlain, one of the Pasteur's collaborators, was also so, and the discovery of the autoclave made possible the discovery of what are now called as viruses. Okay, so Chamberlain's filter was first used in the study of tobacco mosaic disease in the year 1892, when D. Ivanovsky, he was first successfully experimentally demonstrated that the tobacco mosaic disease has been caused by the agent which successfully passed through the Chamberlain filter. So Ivanovsky was the one who successfully showed that this tobacco mosaic disease can be easily filtered using the Chamberlain filter that retains even the smallest bacteria. Okay. So it was an important clue, but contrary to his experimental result, Ivanovsky still maintained that either the pathogenic bacterium somehow passed through the filter or a toxin secreted by them passed through the filter and made the filtrate infectious. So he concluded that it can be either a toxin or some pathogenic bacterium which can be passed through. So we have removed the 40-minute time limit on the meeting. Okay, now, okay, fine. So within 6 years after the experiment of Ivanovsky, Bejernik confirmed by repeating the same experiment. So as I said, till Bejernik it was not known, right? So only after the Bejernik that the same experiment was found, and that the tobacco mosaic disease was caused not by any pathogenic bacterium or a toxin. So before Bejernik, it was thought that it was a toxin or a pathogenic bacterium which was responsible for the disease. But with the work of the Biogennic, we came to know that this is nothing but the virus. So he called them as Contagium vivum fluidum and referred subsequently to it as viruses. So virus means poison. So he observed that the viruses multiply only inside the living cells, but can survive for a longer period in a dried state. You all know that now we have an issue of corona, which is also a virus. So it multiplies only when it enters inside the living cell. Otherwise, it will be inert outside, right? So it will remain for a longer period. In the same way, the coronavirus, as we know, that it can remain in the surfaces for a longer period. But whoever touches the surface, whoever comes in contact with the deceased person, he will get it, and the virus starts multiplying. So it needs a living host to multiply. Otherwise, it cannot multiply. Outside, it cannot multiply, right? So it requires a living host or a cell to multiply. So later on, as I said, it was the work of the yellow fever. Reed and Paul Frosch, they worked on the cattle disease, right? And Reed worked on the yellow fever disease, and it was the fever. So it was the disease of the mosquito. That mosquito was transmitting the disease. So it was prevalent in the African countries, right? So mosquito, therefore they started controlling the mosquitoes, right? It was a vector, it was acting as a vector, and it was carrying the viruses from one person to another person, causing the yellow fever. So that was the first disease to be reported. Why we are referring yellow fever here? Because it was the first fever, viral disease to be reported in human beings. So there are various diseases of viruses in human beings, but yellow fever was the first disease to be recorded as viral disease, right? And it is the mosquito which is the vector that carries the disease. Okay. So thus by the beginning of 20th century, it has been established with certainty that the filterable viruses were independent entities, different from bacteria, and were able to cause diseases. So in the 20th century, people realized that it cannot be called as a bacterium. It is neither a toxin nor a bacterium, but it is smaller than bacterium, and it causes the diseases, an infectious agent, and it causes diseases in plants, animals, and humans, right? So post-discovery contribution, if you look into, after the discovery of the viruses as independent entities, certain important developments were made in virology. In the year 1908, in the year 1911, so viruses responsible for muscle tumor in chickens, right? Discovery of bacteriophages was made. So this was another milestone in the development of virology. So we did not stop there. So we continued to work on viruses, and we came to know the bacteriophages also. So with the work of Ellerman, with the work of Bank, we could know the viral disease. We could know some more diseases of viruses in the form of bacteriophages were discovered, and some muscle tumors in chicken were also discovered, right? So the first published observation suggesting that the bacteria themselves also could be attacked by viruses. So later on, it was Stolt who showed that even the bacteria are infected. It's not only the plants, animals, and human beings, even the bacteria are attacked by viruses. So that those organisms, those groups were called as the bacteriophages. You know that these bacteriophages are nothing but the viruses infecting bacteria, right? So it was Stolt in the year 1915 who showed that the viruses will infect bacterium. They attack bacterium and can result in the bacteriophages, right? So they can cause the disease. So they are called as the viruses infecting bacterium were called as bacteriophages. Stolt observed that the colonies of micrococci and intestinal bacilli bacteria were lysed, and the lytic effect spread from one colony to other colony. So Stolt also observed the colonies of bacterium breaking, they were lysing. So he speculated that the lysis of bacterial colonies could be due to the activity of the virus. So he saw some lysis, some breaking of the bacterial cell, and he called that as lysis of the bacterium, and he found that it could be due to the activity or the multiplication of the viruses inside. Okay, so Todd did not follow up these observations and he remained for Felix D. Harrell to establish decisively the existence of bacterial viruses. Though he could not take any step further, it was D. Harrell who isolated these bacterial viruses from patients with dysentery. So probably caused by the Spigella dysenteriae. So he isolated it. Okay. So therefore, okay. And sorry, where was okay? So he observed that when a virus suspension was spread on the layer of bacterium growing on agar, clear circular areas containing viruses and lysed bacterial cells are appeared. So he observed that when viruses suspension was spread on the agar medium, so he could say clear circular areas containing viruses and lysed bacterial cells were appeared. So D. Harrell demonstrated that bacterial viruses could reproduce only in the live bacterial cells and named these as bacteriophages because could eat bacterial cells. They could eat bacterial cells. They are bacterial eaters. Okay. So establishment of chemical nature of viruses was a very significant contribution in the field of growing virology. Later on, you all know that it was Stanley who crystallized TMV. Okay. So first crystallized the tobacco mosaic virus and found it to be a largely or completely protein. So Stanley showed that this tobacco mosaic virus is caused by a protein. Later on, it was Borden and Peary separated tobacco mosaic virus particle into protein and nucleic acids. Thus, it was established that it is the combination of nucleic acid and protein. So with this work, we could clearly say that the viruses were they are composed of nucleic acids and proteins. So if you look into the history, earlier we were knowing only the diseases. Later on, we could say that they are viruses, and later on we found that this can infect bacteria in the form of bacteriophages. Now we could say that they are made up of the nucleic acids and proteins. This is due to the that intense work conducted by the virologist, and due to the discovery of the microscopes, we could come to this level. So I thank one and all. So let me stop sharing the PPT. I hope you have understood. Okay. So have you understood? So have you understood? Tejaswini, Chiran? Have you understood? Please. So we will stop on this. Okay. 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 Now have you understood? Tejaswini, Chirant, Somdarshana, Sneha, Sushmita, Manikanta, Chirant, Somdarshan, Sushmita, Sneha. Have you understood? Have you understood?
Yes, sir.
Okay. Shall we stop here? Was the PPT clear? Shall we continue with the same app? Shall we continue with the same app?
Yes, sir.
We will continue with the Zoom app only. Okay.
Okay, sir.
Okay. Okay. Are you satisfied with the app? Are you satisfied with the
Yes,
app? Okay. Keep your net
Yes.
properly. Okay. Because it is the net. It's the perfect net that will give you good voice and good video. Otherwise, nothing can help us. Even I should have a good connectivity. Then only I can present. Okay. So thinking that you all will take a very good network connectivity to your mobiles. Okay. Have a good network. Take Airtel or Jio SIM because that will give you wonderful network. Okay. And have a package of 2 GB or 1 GB, whichever is feasible to you. Okay. So I will not come in the way. See that it is not used for other purpose. If you start watching videos, right? If you start watching games, if you start using it for different purpose, the data will come down. So see that it is used only for learning. Mobiles, you should use it only for learning. not for any other activity. If you use it for any other activity, I am sorry. So you will not learn. Okay. Shall we stop here? Shall we stop here?
Yes, sir.
Okay. Thank
Okay,
you. Thank you. Thank you, one and all. Thank you, Chiran. Thank you, Manikanta. Manikanta.
Thank you.
Thank you, Tejaswini. Thank you, Sushmita. Thank you, Somudarshana. Thank you, one and all. Thank you. Have a happy day.
Bye.